Hi everyone, my name is Cha-Cha, and today I'll be going over the amino acids, their structure, and their one-letter codes. I created this channel to try and help keep myself accountable while studying for the MCAT, and as I am a student myself, there may be some mistakes, so I apologize in advance if there are. I'm trying my best not to. Um, so first of all, let's talk about what is an amino acid. So an amino acid, let's talk about its backbone first. So it has that one carbon. All of them have that hydrogen, which allows them to go into space like more easily with just that one hydrogen. And then they'll have the carboxyl group. So, but because this is the carboxyl group, then I'll write the amino group, which will be in red. And this is assuming that this is at a pH of 7. So this plus on the amino group over here, and then the negative charge on the carboxyl group, they will cancel each other out. So amino, it's carboxyl. And then there's always a variable R group, which is what gives the amino acids their spunk. So in a human body, you're assuming that the pH is 7, but technically it's around 7.4. And then one other thing to consider is that there's a buffer for every single pH of plus or minus 1 before it's fully deprotonated or protonated, which is really important with histidine, which can act as an acid or a base. And then this carboxyl group here, because it has that negative charge, this is an acid. And then amino, well, this is just like a broad rule of thumb. And then the amino acid group will be basic because it has that positive charge. So first of all, let's talk about glycine. I'm not entirely sure why the amino acids were named this way. I'm sure there's some um like Latin or something like that. But glycine is G. You just kind of have to remember it. For all the ones that are like super common in nature, they're just like most prevalent in proteins themselves. The first letter of the word will be the one-letter code. So that's true for glycine and for alanine, which just has that methyl group in that variable R region instead of hydrogen. And I forgot to mention, and it's one letter code is A, and I forgot to mention for glycine, this is the only amino acid which is not chiral because it has those two hydrogen groups. In order for something to be chiral, um, it has to have four distinct attachments. So glycine doesn't cut it because it has those two hydrogen groups, and this is important just because chirality is can influence shape and structure and just also how that molecule processes light too. So alanine, and then next to alanine we can have valine, which instead of just having that methyl, it'll have a branch. So at that one carbon, it'll branch out, and this is valine, which is V, and this is the only amino acid that starts with a V as well. Um, and then just going off of that, piggybacking, so that valine had that one carbon that it branched. Leucine has two, and then it branches. So this is one, two. This is leucine, and it is the most common amino acid that starts with an L. And then you have isoleucine, which is chiral, two chiral centers. It's really rare because this and threonine are one of the only, out of all the 20 amino acids, they're the only ones that have two chiral centers. So, like leucine, it has two carbon branches, or two carbons off of the main chain, and then it has the name isoleucine, just iso meaning same. It has two methyl groups or one each that branches off, and that gives it kind of that seesaw shape in a way. So isoleucine. And next to isoleucine, oh, and then here's the chiral center. Pretty much at all at the variable region with that hydrogen, that R group, that's always going to be a chiral center. And then for isoleucine, this. will be your chiral center as well. Um, okay. Let's go for serine. Also, all of these are hydrophobic, and this is serine. Serine is kind of a branching point for some of, oh, why did I do ages? It's not age. Serine, um, it is very similar to cysteine, and cysteine is one of two amino acids that has a sulfur group in it, like methionine. Methionine. So the how you can remember, since I always got cysteine and methionine confused just because they have the sulfur, cysteine sounds very similar to serine. Cis, C, I guess like C and S, they're kind of similar. And they both have just that one carbon, and then they have the hydroxyl group or a sulfur group. And then methionine is really important. Um, I guess these two are really important because they can hydrogen bond, so they're very strong. So they'll have like really high boiling points, melting points, everything that comes along with hydrogen bonds. And then methionine is really important because it can form disulfide bonds, which is an important covalent type of bond that is strong in the MCAT likes to talk about disulfide bonds. Um, so that is something to remember. And then we're almost done with like the hydrophobic ones that aren't aromatic. I forgot to say here, this is proline. Proline is a really interesting amino acid because it forms a ring structure with the nitrogen of the amino group. So it's one, two, three, and you don't really need to know the amino acid structures like super well, unless like you're in biochem, which that's always a fun class. And this is proline. I think, oh, I forgot to, no, I forgot threonine. Oh my gosh. Never mind, I lied. There's one more. Threonine, which is the other um chiral one too. So it's kind of important to know this one. Oh my gosh. So it's one, and then it has that. Okay. Chiral, chiral, chiral. And this is threonine. It's very similar to serine because it just the only difference is that it has an extra carbon CH3 group here. Instead of where serine, this wasn't there. This would be serine. This is threonine. That makes all the difference. So that's our other extra special little guy. Oh, did I do that? Oh my gosh, why is my computer like glitching? Okay. Let's go to the aromatic ones. So why are aromatic amino acids kind of important? So what is an aromatic compound to begin with? So this is our my little benzene. So most aromatic compounds, they have to be cyclic. All of them have to have sp2 hybridization. So that double bond is an sp2. And then it has to have 4n plus 2 pi bonding electrons, but that doesn't really make like why it's super important. So the MCAT likes to test a lot. So these ones will be picked up by UV rays. So around that 250 nanometer like range. And they're the only ones that are like really sensitive, that their electrons will in the sp2 hybridization orbitals will get excited by that. Um, okay. So let's draw phenylalanine. There's that beautiful, beautiful. So phenylalanine, now we're kind of getting into the like naming like one letter codes where it's kind of a little BS. This is a F, like phenyl, like maybe if you have like a list for your like pH kind of sounds like ph a little bit. So that's phenylalanine. And then let's draw tyrosine. So tyrosine is like phenylalanine. It just has that hydroxyl group instead on the benzene. Or, and then tyrosine makes sense just because it has that Y. So tyrosine is the second one. And then tryptophan. I'm really bad at drawing tryptophan. So one, two. Let's see if I can actually draw it this time. I'm sorry for silent. I'm like concentrating. Oh, maybe it looks really wrong. Okay. But this is supposed to be sort of a benzene. Um, so this is tryptophan. Oh, well, there we go. Tryptophan. And tryptophan is very wide. It's a big boy, just like all of the aromatic ones. So tryptophan is W. Oh, I forgot to say tyrosine has a pKa of 10.1, meaning it is basic. So anything above pKa of 7 are like just like on average will be basic. Um, okay. Now let's talk about the on the other side. Oh, let's talk about the basic. Speaking of basic, let's talk about basic amino acids. Basic not because of their structure is basic, but basic because their pKa is like around 10-ish or so. So this will be all of the basic ones will have one, two, three, four carbons, and then they'll have their functional groups. So here's lysine. Lysine special K. This is a very common one. It's not as common as leucine, but hence why it doesn't have the first letter of the name being its one letter code. This is special K lysine. And then you'll have arginine. Three, four. So one, two, three, four. So this is arginine, and it's a pirate. Um, just like really emphasize like the R. Like I guess that's just like the most common one that I can like say. So arginine, it has a NH2, NH2 plus. So arginine, oh, glycine, I forgot to say, its pK is around 10.5. At least that's what my biochem teacher always said. And then arginine is around 12-ish, give or take. And then, speaking of basic, we'll talk about histidine, which is the most, it's not basic, I guess, per se, but it can be an acid or a base because its pKa is around 6, so that's kind of in that buffer zone where it could be protonated or deprotonated. Um, let's see one. I think I drew that right. Wow. Okay. I'm like really bad at drawing some of the rings. I am not an artist. And then histidine is super common, so you'll always see that a lot of times in just like a lot of like chemical reactions. It can like because it could take the hydrogen or give up the hydrogen. So a lot of times proteins will want to have it if there's like something going on, if there's like a chemical reaction going on. And histidine, as I keep on saying, its pKa is around 6. And then let's go on the opposite end of the spectrum. We'll have those acids. So you'll have aspartic acid, which is 1, and then 2. Then it has a carboxyl group. 1, 2 carbons. And then it's twin, sort of. 1, 2, 3. It's older brother, I guess, which has 3. So that's the difference between the two. So this is aspartic acid, and this is glutamic acid. And because aspartic acid only has 2 carbons, so that means that the negative charge of that oxygen of the carboxyl group is more prevalent in it. So its pKa will be even more basic, I'm not basic, acidic and glutamic acid. So it'll have a pKa of 3.8, and glutamic acid will have a pKa of around 4.2. So it's kind of negligible, but if you are like going to be asked like, hey, like which one's more acidic, it'll be aspartic acid because it has less carbons. Um, yeah, and then first one letter code, aspartic acid, aspartic, or like you could think of a T as a D and then it has the acid. So this is a D, then glutamic acid, glutamic acid. I don't know. I guess it's like French or so. I don't even know what accent that is, but it has an E. Um, yeah, and then for our last two, they're just kind of there. You'll have asparagine and glutamine. So asparagine, one, it's the same thing. So one, two, so all the A ones. So aspartic acid and glutamine, I'm sorry, aspartic acid and asparagine, they will both have two carbons since they're A. So A, kind of think of like as less than. A is the first letter of the alphabet, so there will just be less carbons. So this is asparagine, and this is what it looks like. And then you'll have, why is this so far away? And then you'll have its twin or older brother because it has more carbons. One, two, three. It's had more lives, I guess. One, two, three, oxygen. And then this is glutamine, which is a neurotransmitter, which you will hear a lot about in the psych section. Um, and then asparagine has N, and then glutamine is Q. I don't really have much to say about like trying to remember the one letter code. Asparagine, I guess it has an N at the end, and glutamine, the Q is sort of similar to a G, but that's stretching it. So at least for me, whenever I see something where like I don't recognize the other ones, I'm like, oh, that's why. Well, hopefully this made sense. This is just more or less to keep myself accountable while I go through all the chapters and just like kind of the most important stuff on the MCAT. Hopefully you enjoyed listening. I enjoyed, I guess, just talking. But yeah, have a good rest of your day.